We can start. Hello and welcome everyone to our webinar today, tonight, wherever you're dialing in. Our topic for today is our new call for application with AbbVie entitled Complex Human Ex Vivo Models of Tissue Inflammation. My name is Christian Tidona. I'm the founder and managing director of the BioMed X Institute in beautiful Heidelberg, what you see behind. And with me tonight is Tim Radstake and Mark Johnston. Tim, would you like to introduce yourself?
Yeah, hello, good afternoon, good evening. My name is Tim Radstake from AbbVie, Head of Early Immunology Discovery. I'm looking forward to this discussion.
Very good, Mark.
Yeah, so everyone, Mark Johnston, spent the last 20 years in the cell therapy space, and now I'm working with Christian to set up the BioMed X Institute in the United States, and I'm really excited about it.
Thanks a lot, Mark. Okay, before we start, some housekeeping remarks. As you know, in these types of webinars, there is a Q&A box. So if you have questions, just start typing them throughout the presentations and throughout the discussion into the Q&A box. You can also upvote or downvote questions, and we'll then go through those questions either through the talks or at the end. Also, we'll provide further information in the chat, helpful links, and we're supported by our colleagues Rita and Bianca, who are going to make sure in the back that everything is going well. Now, without further ado, I'll start a quick presentation. My part will be, whoops, my camera just went off.
We can still see you. We can still see you.
You can see it? Okay. I have a black screen, so that happens from time to time. It's probably the computer trying to do an update. Okay, I'm back. Very sorry for that. Greetings to Microsoft. Thanks for this. So let me quickly share my screen. I'm going to give you a quick introduction to the BioMed X Institute in Heidelberg, Germany, and then you're going to hear from Tim about our call, the topic of our call at AbbVie, and then you're going to hear from Mark what it is like to do great research in New Haven at our new site close to the Yale campus. So let me quickly try and share my screen. Okie doke. So let's go. Okay, good. Maybe a couple of words about myself so that you understand where I'm coming from. I finished my PhD in virology in Heidelberg. So I'm a scientist by training in Heidelberg, Germany in the 90s. And I always had two hearts in my chest, one of a scientist and one of an entrepreneur. And during my PhD, I already started my first company, which was called Multimetrics. I had a crazy value proposition, 100 tests in 1 drop of blood. Didn't go too well, failed like others miserably, but I learned a lot. Then a cell therapy company. This is where I met Mark. And then after that, I started a stem cell institute called HiStem, Heidelberg Institute for Stem Cell Technology and Experimental Medicine, which is dealing with tumor stem cells. In 2009, I switched to the other side and was the head of the biotechnology region in Heidelberg, coordinating an 80 million public-private research and development fund. And then in 2013, I wanted to do something different, which creates impact for patients ultimately. So the question was, how do we bridge this gap between biomedical academic research and an industrial application that will ultimately help patients? And also my passion is helping research talents to grow. So I combined this passion with my goal and started BioMed X. I'm still the owner of the Institute, and we have a long-term perspective. So we want to create impact 50, 100 years down the road, long time beyond my lifespan. We're currently based in Heidelberg. As I said, for the ones of you, it's a beautiful biomedical research campus, but we're also operating 2 different international sites. One is in Israel called Ion Labs, and our new site in the US, which is run by Mark. You're going to hear a little more about our US site soon. In Heidelberg, we're embedded in the campus of the University of Heidelberg, and we're combining the best of the 2 worlds, academia and industry, and we do this using a new innovation model, which combines global crowdsourcing and local incubation. And we've actually published this new innovation model in Nature Biotech in 2015. How does it work? Whenever we identify a very tough research question with a big pharmaceutical company, so something that would really be a game changer, but there's no solution up to now, We publish this on our crowdsourcing platform worldwide at the best universities and research institutions, and invite early career academic scientists to apply by submitting a very original project proposal, how to solve this particular problem. And usually we get several hundred project proposals from 60, 70, 80 countries. It's really global. And then we sit together with our pharma partners and we select the 15 brightest individuals and we fly them in for a 5-day boot camp where they meet each other for the first time. And then we help them to associate in 5 competing groups and also to combine this collection of brilliant ideas into something we would call a truly brilliant project proposal. So not just a very elegant experimental approach, but also a very solid implementation strategy. And then on the last day, they present in front of the jury, which usually includes the senior management of the sponsoring pharmaceutical company. And if we agree on a winner, this winner is then moved with his or her family to our institute. We organize visa, jobs for their spouses, kindergarten spots, new homes, schools for their children. And they live and work for up to 5 years in our institute, embedded in a major university campus. In addition, of course, this needs some funding. So most of our projects have an annual budget of about 1 million euros or US dollars. So technically, it's a research grant of up to 5 million for 5 years. This is what such a bootcamp looks like. This is still one of the early days. People look still fresh. It's quite an exhausting experience, but it's also very inspiring because you sit together day and night with lots of like-minded top talents from around the world. And with the mentors, you really develop outstanding research ideas, project ideas. And we also continue doing this during the coronavirus pandemic, because we truly believe in the fact that innovation needs intimacy, needs personal contact, needs interaction. The best ideas come during lunch, during dinner, during sports, or pre-discussions. And so we did this during the pandemic, of course, with special permissions from the government, everybody wearing masks, daily testing and so on, but it worked pretty well. Now, the topic of today is a call we published together with our partner AbbVie, and the title is Complex Human Ex Vivo Models of Tissue Inflammation. You're going to hear more about this call from Tim. In the end, what we want is we want to build a completely new research group with top talents from around the world, comprising of 1 group leader, 2 postdoctoral researchers, and 2 what we call research assistants, which are usually PhD students. Now, next slide. What's very important is, since BioMed X is a place where you exchange ideas and help each other, for us, it's very important that everyone is in agreement with our core values. And if you visit in Heidelberg and soon also in New Haven, you will see these core values on every wall, more or less. The first core value is cross-leverage. We inspire and motivate each other to unleash our full potential. What does it mean? We want scientists who are not just scientifically brilliant, but who have embraced the concept of growing by helping others. So sharing their ideas, helping each other to create great new ideas. Second is cross-pollinate. We embrace diversity as it is the source of innovation. So once you visit BioMed X in Heidelberg in Israel, or soon in New Haven, you will find out that every one of us is different, a little crazy, including management, and it's good like that. We embrace diversity. It's very important for innovation that we look upon the same problem from completely different perspectives. And of course, aim high. Our goal is to enable a new generation of targeted therapies. For us, it's very important that everyone who joins BioMed X is on a mission really for, in the end, for the patient, but also for him or herself to make most out of these up to 5 years and get to the next big career step in this limited time period. In Heidelberg, we've been working with 9 different pharmaceutical companies and also with our incubator in Israel. And with these companies, we have started 20 research groups so far in the past 10 years. Each of those groups with about funding of about 5 million euros on average. In addition, we have started 4 startup teams with the very same model in Israel at Ion Labs. I'm going to explain also a little later. And also we publish. It's very important because some of our fellows that join BioMed X, they don't want to switch to industry or into startup. They want to go back to academia. And it's very important that you maintain a very attractive continuous publication record. So we also publish in peer-reviewed journals, so far more than 50 publications. And we also educate. We have more than 60 bachelor, master, and PhD theses, which were completed at the BioMed X Institute. Our focus in Heidelberg is deep disease biology and artificial intelligence. Going a little more into detail, almost all of our teams are working with primary human materials, building human organotypic disease models, for example, in different disease areas, which is very important in our view at the moment, because so far we're quite good at curing mice, but not so good at curing humans. So it's very important that we try to model the actual human tissue as closely as possible, especially including things like the immune compartment, where immune cells move through organotypic tissue microenvironment. Also, what's very important is all our teams generate very deep multi-omics data sets. So they need bioinformatics, data science, artificial intelligence, and machine learning to make sense out of all of these data. And also we have teams that are focused on sensor development, optical, electrical sensors. And you need to keep in mind at BioMed X Institute, everybody helps each other. So they all communicate with each other and they all help each other to meet their goals in their particular research project. A couple of words about Ion Labs. We started a little more than 1 year ago. It's a collaboration between Merck, Pfizer, Teva, AstraZeneca, Amazon, and an Israel venture capital company. And we are close to the campus of the famous Weizmann Institute in Rehovot. And with our model, we are generating startups in Israel that focus on artificial intelligence for drug discovery and development. In this case, we have multiple pharma sponsors per project, and we have enough funding to generate 20 startups within 5 years with this particular model to solve all kinds of problems with AI, from early target identification to late-stage clinical trials. We've started our first 4 startups last year, and now we're publishing a call every quarter until we have 20 startups. And of course, our new baby, new kid on the block is our new site in New Haven. You're going to hear more about from Mark, close to Yale University, a fantastic academic science environment, and also a great place to live. Now, why should great talents come to BioMed X? First of all, it's a fully funded 3 to 5 year research group. So our group leaders usually, they don't need to have leadership experience. This is what you learn at BioMed X, but you get quite a significant grant of up to 5 million. And then, of course, you focus on outstanding research and publications, no teaching obligations, no other tasks. You just focus on your own research project. It's a very creative cross-cultural teamwork atmosphere with people from around the world. You get guidance from experienced mentors from academia and industry. You're meeting your mentors from academia and also the sponsoring pharma company once a month and discuss past and future experiments. You get access to materials and data from your pharma partners. So it's quite a unique situation. It's an outstanding environment with direct access to leading research institutions and companies. In New Haven, as you know, Yale University is one of the top universities, but there's also lots of core facilities, other institutions, startups with whom we will interact and collaborate. You will get leadership and entrepreneurship training. And of course, what's very important, it's BioMed X is a risk-free assessment of a possible future career in a startup or big company, because as I said, we publish. So you can always go back into a tenure track and become a professor if you don't like industry or startup. Can we attract top talents from around the world? I think after now 10 years and 300 employment agreements down the road, I can say, yes, we can. This is just some of the logos where people came from before they joined the BioMed X Institute. Where do they go? About 50% go to industry 6 months before the end of the term. We spend a lot of time with each of the fellows before they leave, helping them to polish their CV, to find out what's the next best step in their career, and then opening up our Rolodex and making connections to their next employer. We have quite a few of our early alumni now who are in very senior leadership positions in academia or industry. This is our centerpiece, career.bio.mx. This is what we call our career space. All our jobs, all our calls for application are published on this platform. Once you're registered and you opt in to get information, you will get an email whenever we publish a new call for applications. Either in Heidelberg, in Israel, or in the United States. and you can apply with the very same profile as a matter of by a simple click. And of course, you're very welcome to follow us on LinkedIn. Our main platform is LinkedIn, and stay connected with all of us. And with that, I stop my presentation and hand over to you, Tim.
Thank you, Christian. And let me start to share my screen. Just a second. Always takes more time to share. Where's my share button? Here it is. Can everybody see my screen? So if I don't hear anything, so wow, thanks, Christian. Thanks, Christian, for the mesmerizing introduction, which I think, and I'm quite sure actually, this is going to be a very exciting project. So indeed, I'm going to tell a little bit more about AbbVie, AbbVie Immunology particularly, and then of course about the big ideas we have building these complex in vitro model systems. But first, before I do so, I wanted to explain a little bit about myself, because I guess it also gives you a little bit of taste why I actually joined industry. So I am an MD, so I actually studied medicine in Nijmegen in the Netherlands. I did some extracurricular activities. I was very much and very early in science. So I spent 6 months in Paris in Geneton, the Human Genome Project, did my PhD in 2003 and became and fell in love with immunology. I did my thesis on genetics as well as dendritic cell biology. And then I started my residency in rheumatology. So did my postdoc in Boston. That's why I fell in love in Boston and Cambridge with Robert Laviadis on systemic sclerosis, so mainly about fibrosis. And then I became a full professor in rheumatology and had a lab in translational immunology in Utrecht, the Netherlands. So that was actually the journey really in clinical. I always had translational research besides. So I did clinics in the morning and saw the problems that I wanted to translate into medicine and helping as much as many patients possible. In 2017, I started a unique program in GSK, which is called the Immune Catalyst Program, where I really started to work in industry. Although I was an academic scientist, I learned a lot of there. And I have to say also that most of my grants, I had a big group, about 55 people, were with pharma. So I have a lot of experience in working in and with pharma, and actually started the stealth mode company on one of the targets that we identified back in the Netherlands. But so I had a big lab, had quite some interesting and high impact papers, had a lot of funding. But still was doubting my impact. So then when AbbVie came along and said, well, you know, Tim, are you interested to join pharma? I made the choice to do so. And so that's how it became in clinical development, where I started in AbbVie, and I started as a medical director. So AbbVie has 5 big pillars: immunology, that's what we're going to talk more about today, but also oncology, neuroscience, ophthalmology, research, and aesthetics. And we are separated around the globe, basically. I'm showing our US areas here. So at the East Coast, our headquarters is Worcester. It's about 40 miles out of Boston. That's where our global biologics organization is, operations and science and technology. Also a small part of our immunology franchise. But the biggest place for our immunology folks is actually Cambridge, which is divided in immunology and neuroscience discovery research. Headquarters in the US is based in Lake County near Chicago. That's a huge campus. I think there's about 25,000 people working over there. And then we have 2 smaller sites, Irvine, which is our aesthetics and eye care. And then more recently, our oncology discovery research in South San Francisco. Cambridge is quite close to New Haven. It's about 2-hour drive. So it's perfect for us to really start up a really interesting collaboration. So as a rheumatologist, I actually saw the tremendous impact our medicines had on patients with rheumatoid arthritis. So in the picture below, you see a patient with rheumatoid arthritis before TNF inhibitors and all other medicines that we currently have in our arsenal. And on the top picture, you see where we are now. So from a total bone loss, total incapability of using your hands towards a disease that you can quite well control for patients with rheumatoid arthritis. And so this happened in many other diseases that we're now touching with our medicines. But if you really look at it, and this is a slide from our own internal data and shows, okay, what's the disease activity in patients with rheumatoid arthritis and where do we really impact? And you can see that the 2 lines here is where we want to be. It's low disease activity and complete remission. And you can see that the majority of patients are falling below that. So there's still much to do, not only in rheumatoid arthritis, but in autoimmune or immune-mediated inflammatory diseases in general. So about 2 years ago, we set our new vision, which is really driving a cure in these patients. So our aspiration is really, can we define the new standard for therapies? And we defined also how a cure looks like. So where we are now treating clinical symptoms, we really want to have that step up towards clinical remission and then functional remission and maybe even a molecular cure where people might not even need therapy for prolonged periods of time anymore. But the problem is that drug discovery is really difficult. And just to take you over a couple of complicated slides, but the main message here in every part of this drug discovery, which you can see on the right, is where we start from target validation and identification, but also up till phase 1, 2, and 3 clinical trials. You see that there's roughly 90% of attrition over time. And if you really look at it, it's lack of clinical efficacy, but also toxicity and drugability issues. So if you really look at it, about 90% of the failure is in the hands of discovery, something that we need to change. So what drives that lack of efficacy or attrition rate is, I believe, and that's where we really start to work on, is the lack of disease biology. So it's a lack of understanding of disease biology, but also other things like how do we prioritize and how do we benchmark or differentiate our early assets. Model selection. You talked a little bit about mice models, Christian. We all know that we can cure diseases in mice, but that's, of course, very different in humans. So what's the best model to really drive early decision-making in drug discovery? And I can give you 2 examples, actually, is why this is difficult. So on the top, you see a patient with psoriatic arthritis, which is a patient there you see arthritis, so joint inflammation on the left and the skin problems on the right. So this is actually the same patients with 2 sites of locations. But then if you look at steroids and TNF inhibitors, IL-23 inhibitors, you see different sizes of effects, which makes it very fascinating that in the same patient, you don't see the same response in those different regions, telling us that there must be different molecular pathways driving these diseases in those different spots. The other example is systemic sclerosis, which is a very heterogeneous disease. It's a disease where fibrosis in the skin and internal organs leads to disability, severe disability in patients. And what happens actually is that there's a lot of failure in moving from phase 1, phase 2 to phase 3 clinical trial. And this is just a slide from the literature where you see that if you look at molecular profiles in these patients, or a different subset of these patients, you see different immune profiles. So what happens is that those early phase, in this case a phase 2 trial, is really focused around specific molecular fingerprint because it focuses around a particular subset of patients. But then what happens in a bigger trial, like a phase 3 trial, you have to expand in multi-locations, potentially multiple countries over the world. And so you're missing out on that initial molecular profile. And that's potentially why a phase 3 trial fails. So again, we need to understand disease much better than we currently do. And I think this is a perfect example of what happened the past 10 years when IL-17 and IL-23 got identified in psoriasis as the main driver of disease. And at the same time, it was identified that it's not only those 2 molecules, but also that the role of T cells and keratinocytes. And of course, drugs were made against these targets like anti-IL-17, IL-23. And where the field has now progressed is having more than 90% of patients with psoriasis have almost no symptoms. So where psoriasis was 10 years ago, it's where we are for all the other autoimmune diseases at this point. So we have to push the needle on this. How we think we do this, at least within AbbVie, and what you see around us, also in academics as well as in pharma, is to build on that molecular understanding of disease. So internally, we use our clinical trial samples as well as academic collaborations, deep molecular profiling to get to those fingerprints of disease, but then also have a couple of other methodologies developed to really identify the best potential targets early on in that drug discovery program. So I will not talk about the 4 on the right. I will only lift out the immunized model systems because that's the topic of the call. So we have been building on this internally, and what you see on the left is a simple 2D co-culture system where we could measure different stimuli on, in this case, keratinocytes, a co-culture of fibroblasts and PBMCs. And we can measure different cytokines and the response of internal assets and external assets, and make decisions. Okay, what does look better, what does look worse? And on the right side, you see that there's progression into what we call 3D organotypic model systems. In this case, you can see that there's pathology mimicking what we see in patients in spongiosис. So it's a combination of the epidermis and dermis, and you can start to measure different cytokines, different stimulations. So although this is a really great progression as to what we can do in vitro, it still doesn't mimic human disease by far. So what we are really driving internally, and hopefully also with this really interesting collaboration, is to build a chip or disease on a chip or a complex in vitro model system. Doesn't really matter how we call it, but what's as close as possible to a human being. And so how that is going to look like, we don't know yet, but that's where we need you to come to our team. It could be the combination of multiple immune cell subsets with vessels, with extracellular matrix, what have you. It just needs to be as close to human beings as possible to make that final choice, what is entering the clinics. So we do have a fantastic team already for you. This is the team that will be liaising from the pharma end to BioMed X. So we have Sarah Wilson, who is covering the gastroenterology. We have Vanessa Morales-Tirado, who is covering the dermatology. And we have Anna Jarolina and Marianne Kleiman, who is covering rheumatology. Anna is more on the rheumatology side, on the rheumatoid arthritis, synovium, and Marianne is covering vessel on the chip. So we have a lot going on internally, but still felt very clearly that we have to drive the future by building out with BioMed X. And what would be nicer to really start that off with you in New Haven. I leave it here. I'm open for questions and give the word to Mark.
Great. Thank you, Tim. Appreciate that. Let me start my screen. There we go. All right. So everyone, I'm going to talk about, living and working in New Haven, Connecticut. First, I want to do my life, my work, my passions. This is an exercise that all new staff at BioMed X do to quickly describe ourselves to the team. And one of the what I'm really passionate about is work. If I'm not passionate about it, I won't do it. And I think one of the things that attracted me most to BioMed X are the really cool, fascinating projects that we work on. If you've ever gone to our website, you can see the diversity of the projects, and everything's cutting edge, everything's novel and groundbreaking, and being part of that is really rewarding to somebody like myself. And with that said, family is the most important thing to me. It's taken me a while, but I finally figured out how to have a good work-life balance. I'm a big lover of travel. I've lived in big cities, London, Barcelona, New York, Los Angeles. So having a lot going on is really important to me. And I'm also I'm a fanatic of exercise. I like to hike, I like to bike, I live near the beach. We're going to the beach every weekend. And, when Christian and I started talking about where we're going to place the BioMed X Institute in the United States, to be frank, New Haven, Connecticut didn't register really at all. I thought it was a sleepy college town. And then I visited and I was very, very wrong. What I found was a vibrant biotechnology ecosystem, much larger than Yale. Yale is, of course, central hub of the city, and having all the benefits associated with being adjacent to one of the top medical schools in the world. Yale's been around for over 300 years, and you really feel a deep respect from the city to the university, and you can see it, you can hear it just by entering the town. We're, as Tim said, we have a close proximity to Boston, so we'll be very close to our pharmaceutical partners like AbbVie, and then really to top it off, it's really, really low cost of living. It's very reasonable, and quality of life in New Haven and the larger Connecticut it's exceptional. I'll talk about that in just 1 second. But staying on the topic of the ecosystem, so New Haven has Yale, New Haven has pharmaceutical companies and biotech, but the state of Connecticut has a lot of pharmaceutical and biotechnology companies. And this over to the right, you can just see a very, very small sampling of the number of biotechs. It's the number 3 most educated workforce in the United States. Why that's important to you is you can source local, high quality talent for your team. And then BioCT is just one of the groups focused on increasing life sciences in the state. They've negotiated discount pricing for services and equipment. They have monthly networking events, seminars, and they're just one of a number of groups that are promoting life sciences within the state. We're also, supportive. We have a very supportive partner, Yale University. You get mentoring by top, Yale academics. You get access to some of the best services and equipment in the world. There's a CyTOF department. There's a lot of mass spec on campus. You've got confocals with multiple lasers for enhanced 3D imaging. So you will have access to this really cool technology that's going to be walking distance from the facility. You'll be able to engage with Yale's postdoc association to help hire high quality members of your team, and you'll be able to network with some of the leading scientists and researchers in the world. Beyond New Haven, you have high quality of life in Connecticut. It's for education, it's number 3 for K through 12 or high school education. Number 5, quality of life in the United States. It's the number 4th lowest crime rate in the US. Great place to raise family. And for me, 618 miles of scenic coastline and water and hiking and biking, these are really, really important for somebody like me. And I think that you will also enjoy that. It's also, very, very international city. The number of restaurants, there's something like 550 restaurants within a 5-mile radius of central New Haven. Any kind of food that you want is there. There's plenty of bars and clubs, regional theater, sports teams, anything that you actually need. And if you're bored and you want to get out of town, you're a 5-minute drive to New Haven's regional airport, 40-minute to the international airport, and you're only an hour and a half to New York and 2 hours to Boston for all of the theater and sports and nightlife that they're quite famous for. As I mentioned, camping and beaches are only 10 minutes away. So New Haven is considered the cultural capital of Connecticut. And, the takeaway for me is that New Haven has all the benefits of a large international city while feeling like a comfortable village. I love it. And I know that you will too.